Yes.
Great. All right. Well, welcome to today's N3C Community Forum. Today, we're going to hear about incorporating external real-world data into analysis of randomized controlled clinical trials, bringing real-world data and clinical trials together. And this is a really hot topic. I actually know that I was just on a panel with the NCI Cooperative Group, ECOG Akron, on this exact topic. And so this is really extremely timely. So I'm personally quite interested to hear this, and I think we could have a really good discussion. Next slide, please. So then next week, we're going to hear about the use of cardiopulmonary exercise testing in addressing issues such as fatigue and pediatric PASC by Dan Cooper. The 23rd, outcomes of COVID-19 in people with hemophilia, along with the monthly Recover Long COVID project team updates. May 30th is Memorial Day. June 6th, protocol for treating long COVID patients that either completely resolves or significantly improves the symptom burden. The 13th of June, BARDA challenge updates, and no presentation on July 20th due to the Juneteenth holiday. Next slide, please. So, okay, so we're up to 4,999,630 cases. That's quite something. And actually, at the real world data cooperative group presentation, I actually talked quite a bit about N3C as a best practice mechanism for doing some of these things. The concept of an enclave, obviously not new, has been really really remarkably well instantiated here. Next slide, please. Okay, so Lillian, do you want to share your screen?
Yeah, sure. All right, so can everyone hear or see and hear me, the slides?
Looks great.
Yes.
Okay, great. Thank you all. So yeah, a bit about me. I'm Lillian, aka Lily, like the flower, Hayne, and I am a PhD student at the University of Minnesota in the Division of Biostatistics, and I am a tracked TL1 scholar. And I'm going to be presenting a little bit about kind of the first chunk of my dissertation work, which has been on developing a statistical method for incorporating external real-world data into randomized controlled trial analyses. So kind of just how we're going to walk through this wonderful world is we're going to start with just a little bit of background and motivation, kind of like why does it even make sense to borrow? When is it applicable? Some current borrowing approaches in the kind of current stats literature that people work from and the different frameworks for that. The proposed approach, which I'll kind of outline some motivation on how we're working with real world data or non-randomized data instead of trial data and theory behind it. And then an application to a flu randomized controlled trial, kind of a reanalysis for getting a little bit more precision within a subgroup analysis for a flu RCT. Just a summary, and then some future work, which I think will hopefully be kind of the discussion with this group where people are maybe inspired to kind of find different ways in which the statistical approach could be applied to your own work. So kind of the big question that we're looking to answer with this is kind of how do we make trials more efficient? We saw this with kind of COVID and this amazing enclave, this N3C data source kind of coming together really quickly and really efficiently as a source of data. And we're kind of at this point asking the question from a statistical perspective in terms of how do we make trials more efficient after they've already been completed in the analysis stage? Like how do we use stats to actually make these trials get as much out of that time, money, and kind of participants that we can? That's kind of the big question that we're asking here. So a bit of background, as I'm sure kind of everyone knows and maybe loves, RCTs are really the most rigorous way that we have at this point to kind of find unbiased causal treatment effect estimates. And the beauty behind these randomized controlled trials is this kind of, one of the beautiful things is this randomization, where you randomize people to either a treatment or a control arm. And what that effectively does theoretically is you're kind of shaking out all of the measured and unmeasured confounders, or all of measured and unmeasured differences between the treatment and control arm. And so when you finally at the end kind of compare the two groups, the only thing that is really different is that treatment effect. So the treatment that you kind of gave to the treatment arm. However, they do take a lot of time, money, and participants to kind of set up these distinct trials. And one way in which they're kind of often criticized for being a little bit inefficient is that they don't use really kind of effectively any outside information. So one kind of what seems like a easy or low-hanging fruit or concept to improve trial efficiency would be to supplement the trial sample size with some form of external data. And what I mean by kind of external data is really anything that's collected outside of your current clinical trial. So this could be, hopefully, kind of ideally, it would be previously conducted RCTs, or randomized controlled trials, that you have that randomization, it's from the same target population, and you can kind of feel more comfortable really using those earlier phase trials. But we also have all of these other sources of data. We have observational studies that are often kind of plentiful and sometimes even being conducted at the same time as the randomized trial. And then you also have kind of real world data, or electronic health records, or I think N3C data as well, which is kind of constantly being collected and is a really rich, 4 million cases, that's a huge source of data that we could potentially, hopefully, leverage to make our trials a little bit more efficient. So kind of why don't we already do this? What's the problem here? We would think we have information at our disposal. Why don't we just take everything, pool it all together, and then just figure out the best, kind of most efficient estimate for treatment effect? And the big reason is, as I'm sure everyone already knows, bias / confounding. So when you're kind of naively pooling different sources of data into your trial, you're now really kind of ruining that internal validity, or that randomization component. And you can no longer have a truly unbiased treatment effect estimate, because you could have measurable kind of differences between your treatment arms. So it's kind of some sources of bias, and I kind of camp these into measurable differences and kind of unmeasurable differences. And one of the major sources of bias, especially when you're using something like external kind of real world data or observational data, is kind of measurable population differences. So in the trial, you're usually targeting a very specific population, say people who are between 18 and 45 years of age, and who are non-smokers, who have some pre-existing conditions or no pre-existing conditions. And in like a real world data or an observational data source, you often have much broader inclusion-exclusion criteria. So you're no longer kind of targeting the same population between the two studies. And oftentimes what you'll have for kind of an external data source is you'll only have data on a control arm. So if you have only untreated people, and you all of a sudden incorporate a bunch of people who are very different from your trial population into the control arm, you're clearly going to have very different groups of people that you're trying to compare, and you no longer have an unbiased treatment effect estimate. So that's kind of some examples of measurable population differences, and why we don't just want to naively pool everything from external data into our trial. Some unmeasurable population differences, there could just be something unknown. Maybe your external data source is from the kind of 90s or something, and your trial data source is currently being collected in like 2020 or 2021. And we got just better at kind of treating this disease, or all of a sudden everyone started drinking more water, which turns out it helped in that context. And as such, your kind of external data source is no longer similar to, on an outcome level, your current trial data source. And in that case, again, you kind of might not be able to measure it. There might just be something unmeasurably different that you didn't capture when you're measuring something that is creating differences in outcomes. So if these differences are substantial or create kind of a prohibitive amount of bias, just too much bias within the outcomes, then we can kind of think of your external data and your trial data as no longer exchangeable. And we, in that context, kind of want to avoid borrowing from your external data to supplement your trial. So how do we kind of figure out how much we want to borrow? And how do we currently borrow if we do? The idea of borrowing from kind of external data is really not new. There's a lot of research on borrowing, and even in the 70s, kind of originally, Pocock, who is a very famous statistician, outlined 10 acceptability criteria for kind of external data to be incorporated into a kind of historical control arm. And these are in the figure kind of to the right. And these are pretty stringent, or some of them are in the figure to the right. So these are pretty stringent. You have to have the same treatment as the randomized controlled trial. So if you have like anything that's even varying within your kind of treatment regimen, which is commonly the case, especially in a real world data context, it's maybe even entirely different. You have to have everything be in kind of a recent clinical study. You need to be using all of the same sites, and you need to be fairly sure that like nothing is different, which is kind of an impossible thing to be sure of, because we can always have something that is unmeasurably different between these populations. So there are some more kind of fun, a little bit fancier statistical approaches that are kind of more recently developed that usually kind of downweight or discount the external data in some way to kind of mitigate the introduction of bias. So you're taking your trial as being kind of the kind of primary or more important source, and you're downweighting or discounting kind of your external data source a little bit, just to make sure that it's not introducing too much bias. And there are kind of two major ways in which we can downweight or discount your external data. One is static, where you kind of define the degree of borrowing before you've even seen the data. So it's like a clinician or some sort of expert maybe will say that the data is 50% as similar as to the trial that you're working with. So you kind of choose that number based on kind of information at your disposal, or maybe it's that you know that the target population is slightly different, or you're trying to use an adult to a pediatric population. You have some information on how similar this external data would be to your current trial. And that's a little bit limited because you can't necessarily account for if they end up being really different on an outcome level for some unknown reason that you didn't foresee. Then you would still probably borrow the same amount because you're defining it kind of before you see the data. And the second approach, which is a little bit more kind of fancy or fun, I guess, is the dynamic approach, which we'll kind of hone in a little bit more on today, where the degree of borrowing is determined based on the data. And there are many different dynamic approaches that are out there. The one that I'll kind of talk about more today as an example is multi-source exchangeability modeling, because this is part of the kind of approach that we're taking with the MEM step of the SS mixed MEM that I'm kind of breaking down today. So the general idea behind multi-source exchangeability modeling is that you work out what happens under each exchangeability assumption. So if you have one external data source and then one primary trial data source, you see what happens when you fully borrow and you assume that those two sources of data are exchangeable, and then you see what happens when you only have the trial that you're working under and you assume they're not exchangeable. And then from there, you kind of weight these two results based on how similar the data looks in the end. So you would avoid borrowing if you have very different outcome information between the two data sources, and borrow more if they appear to align, or if they appear to be kind of exchangeable. So kind of the pro of this dynamic approach is if there is some unexpected or unmeasured kind of uncapturable difference between the data sources, you're still able to kind of avoid borrowing in that context to hopefully mitigate bias from using external data. So kind of what do these two approaches assume? So generally speaking, these current kind of borrowing approaches assume that we have external trial data in the same or similar populations as the current trial. So if the populations are really different, they're going to avoid borrowing and be pretty inefficient because that's probably going to impact your outcome. And in the dynamic borrowing approach, you're all of a sudden going to be in the case where they're just not exchangeable and you're never borrowing, even though potentially there could be some group of people that you could borrow from the external data. So we kind of begged the question of what if we don't have any previous trial data, and if we don't have any previous trial data on your target population, especially to borrow from, how can we kind of effectively figure out a way to borrow from this external kind of non-randomized data in some way? So when you're borrowing from real world data instead of trial data, we can kind of think of two real new sources of bias that kind of need to be accounted for. So One would be kind of population heterogeneity, where your external data source and your trial data are from different populations. So this is kind of what I alluded to with the measurable differences between the populations. So one example is the inclusion criteria between the two studies might be very different, but we still want to figure out a way that we can kind of leverage the subgroup of people that is similar from the observational or external data into the RCT. So we're trying to account for or figure out a way to account for these differences between the two studies. The second is unobserved residual differences between studies. So you could even have kind of this, again, this unmeasurable, maybe even uncapturable differences between your external data source, your real world data, and your trial data. And one example of that is kind of that treatment drift that I talked about a little bit earlier. So we really want to account for and mitigate bias from these two sources in this new context when we're trying to leverage external real world data as opposed to trial data. So that kind of motivates our SS mixed mem approach that I've been talking about a little bit through the course of this. Step one with the SS mixed step separates our real world data source or our external kind of non-randomized data into two groups, a non-representative subgroup and a representative subgroup. And what this step is we're kind of honing in on the people who are similar to the randomized controlled trial from a measurable kind of baseline covariate standpoint, like who is it that maybe would have enrolled within the trial. And then from there, the second step is the multi-source exchangeability modeling step. And with that, we use kind of the MEM dynamic borrowing framework to figure out or fill in this kind of gray question mark in terms of how much should we actually borrow from an outcome standpoint from this representative subgroup. So that's kind of the two general idea behind the approach is step one, separate the real world data into your non-representative and the people who are similar to the trial. And then step two, figure out how much from this representative subgroup we should actually use to incorporate into the RCT. So our assumed setting is we have a trial that's already been done, and then we have an observational study, and we're kind of making these big assumptions that we have equivalently defined binary outcomes. We have a covariate matrices for both studies with equivalently defined baseline covariates, and we want these to be measured in both studies, and we assume that we're in the complete case scenario. We'll let S be a study indicator, so when S is 1, you're in the trial, and when it's 0, you're not. Your observational study is untreated. So in this context, we are only supplementing the control arm, but you can extend it to supplement both if you have maybe a subgroup of people who are treated, but potentially not treated in the same way as the RCT. So you could have differential borrowing between the two arms. So our 0th step, or step 0, is the propensity score step. And with this, we're kind of looking to, as is commonly the case in causal literature, kind of mimic some of the properties of a randomized controlled trial. And the propensity score does this kind of by balancing the balancing property of the propensity score, where it balances baseline distributions between two arms. So in this context, we're looking to kind of hone in on the people who are similar to their RCT. So we're looking to balance the distribution of baseline covariates between the trial and the observational study. And as such, we model or define the propensity score as the probability of enrolling in the trial based on all of the kind of baseline covariates that you have at your disposal, or those that are at least kind of prognostic covariates and the ones that you know are related to your outcome. The next kind of step 0B is we normalize the propensity score with the Logit transformation for our SS mix step. So for step 1, the SS mix step, what we're doing is we're using a semi-supervised mixture model on the propensity score. And what this step does is it draws from that representative subgroup of the observational study that has the same distribution of covariates as the trial. So we assume that the Logit transform propensity score for the RCT is from a normal distribution with a mean and a precision. And then we assume that the Logit transformed propensity score for the real world data is from a mixture distribution, a 2-component mixture distribution. And what this is we're trying to kind of capture the people from component 1, or the people who are have propensity scores that are from the same distribution as the RCT. So how we do this is we have Z is defined as a latent indicator of component membership, and it is known to be 1 in the RCT and unknown for the observational data. So we're trying to capture essentially the people with Z equaling 1 from the observational study. How we do this is we use a semi-supervised mixture model on Q, and we get this kind of a draw from the representative subgroup from the real world data from the observational study that could potentially, hopefully, be incorporated into the RCT. So kind of the general idea within the visualization framework for this step is that the orange people are the randomized controlled trial participants, and then the real world data is composed of kind of two people, those who are like the RCT or the orange people, and then those who are not. And we're trying to hone in on or pull out the people who are from the representative subgroup via this first step, this SS mixed step, and get this representative subgroup, which is people who are more similar to the RCT based on these measurable characteristics. Our step 2 is we then use this representative subgroup as our external data source in the multi-source exchangeability modeling framework. And what we're doing here is we're using the outcome to effectuate kind of an appropriate degree of borrowing from this representative subgroup or from a draw from this representative subgroup. And the idea here is that we have kind of one representative subgroup. So we have two exchangeability assumptions. You kind of work out what happens when the representative subgroup and your RCT are exchangeable, and then you work out what happens when they're not. So when E equals 1, the event rate in the representative subgroups in the trial are assumed to be the same. When it's 0, they're assumed to be different. And then the exchangeability indicator kind of is updated based on the data through a posterior probability of exchangeability, and the control arm posterior is a weighted average of these two assumptions. So we'll see this in kind of like a little bit more clarity standpoint if we put a beta prior on your kind of event rate in the RCT treatment arm, and then a beta prior on your control arm as well. And then you let the number of responses and the sample size of the treated arm be RT, so T is treated, RC is C is control, and then R sub R is representative subgroup in this context. So since the event probability for the treated arm is estimated using just the trial data, we're only using the RT and NT for the kind of posterior for the event rate in the treated arm. And then for the posterior in the control arm, we're using this kind of weighted average where we weight the different components, or the one where we're exchanging that and using kind of the RR group in the beta posterior, weighted by how similar they are. So it's kind of a function of the similarity of the outcome of the data. And then you can also, if they're very similar or the representative subgroup has a really different response rate than the control arm, then we would kind of weight the component where you're only using the control arm individuals for the event rate posterior kind of more heavily. So it's kind of this function of the degree of similarity between the representative subgroup and the control arm outcomes. So kind of this visualization again is you take this representative subgroup and you have your randomized controlled trial, and you're using this MEM step or using dynamic borrowing to kind of facilitate how much to borrow from this representative subgroup. So we did kind of a real data application to a flu randomized controlled trial. And this was conducted through the International Network for Strategic Initiatives in Global HIV Trials, or the INSIGHT network. And it was the flu IVIG RCT, which enrolled patients from 2014 to 2018, and it was a double-blind placebo-controlled RCT. And then there's also a really cool kind of observational cohort study called Flu 003, which has been ongoing since 2009. And it's a cohort study on hospitalized individuals to kind of characterize outcomes in individuals hospitalized with influenza. The inclusion and exclusion criteria are not exactly the same across the two studies, kind of as anticipated. The RCT has a little bit more stringent inclusion-exclusion criteria than the observational study, which kind of takes whoever is there. And they were both kind of conducted via the INSIGHT network. So a lot of the same sites were used and a lot of similar covariates were measured. So this is kind of a really wonderful application for this approach. So we have a binary indicator of hospitalized or dead. So we're in the kind of binary outcome setting, which is why I used that for our kind of motivation. We have 308 individuals from the flu IVIG study with 156 treated patients and 151 in the placebo. And the flu 003 study was composed of over 1,000 individuals. So the flu 003 data is untreated, so we would only be eligible for incorporation into the control arm of the RCT. So we are in that context where we're again only borrowing on or only supplementing the RCT control arm. And our goal with this approach is to incorporate kind of some of the observational data into a subgroup analysis of the flu IVIG by influenza subtype because there appeared to be some sort of signal of a treatment effect heterogeneity so that we can get a little bit more precision within this subgroup analysis. So the Logit transform propensity scores. So we just did step 0. We got our propensity scores and we got it via glider, which is kind of a grouped lasso approach. So you can do some form of variable selection as well to figure out which kind of covariates are actually related to the outcome as well as to the kind of are different between the studies. So as we can see, what's really nice about this is it does look like our Logit transform propensity scores for the kind of flu 003 study are from a 2-component mixture distribution in some way. So it looks promising in that regard. And then for the kind of SS mix step, we can see that we are at least pulling our propensity scores kind of closer together. So this is the overall flu IVIG study as the farthest left column, the flu 003 study for all of the people that I had available after cleaning a little bit, which was 1502, and then the flu 003 study with people with Z equaling 1. So those who are determined to have propensity scores that are kind of from the same distribution as the RCT. So the propensity scores are being pulled closer. And we see that this also kind of really impacts the kind of new score, which kind of gets separated into those that are a lot lower and then those that are higher. And then the kind of indicator of Black race, which also gets kind of a lot closer between the two groups. So we are pulling people that are more similar, at least in some way, to the flu IVIG study. And we do see that with subgroup A, we are gaining kind of some precision with our treatment effect estimate with an effective supplemental sample size, which is just kind of a measure of how much precision we gained by borrowing, comparing to the reference, which is just using the trial data. And we see that these are kind of well above 0. And as such, our confidence intervals can't really tell, but they're a little bit more narrow, and that we are kind of definitely still in kind of the null case, but at least we can say with a little bit more certainty that it seems like there might not be an effect. For influenza B, similarly, we are again kind of pulling the people that are more similar to the RCT based on propensity score values as well. So we're really separating it into kind of the propensity scores that are more similar when we pull the right, like the component that we're looking at. And this similarly impacts the kind of Black race percentage, as well as the enrollment location being the ICU. It kind of really makes it a lot closer to what looks like the RCT. And then, unfortunately, I think in this case, maybe not unfortunately, but in this case, it seems that there is kind of a difference between the outcomes, or some sort of unmeasurable, uncapturable difference between the kind of influenza subtype B and the RCT individuals with subtype B, as we're not really borrowing in this context. However, the kind of odds ratio was so stark, it looked like it had such a big signal within the RCT that it does seem to still kind of be getting the same results, but we're not really getting that much from borrowing. So kind of an overview of the approach, we kind of motivated and developed the SS Mixmo approach to incorporating external observational data into RCT analyses. In simulations, it appears to be performing well. We're effectively kind of leveraging a subgroup of observational data while mitigating bias. And in an application, we kind of saw how this approach really would work, where it does kind of hone in on people who are more similar based on those covariates included in the propensity score. And then it does kind of borrow from those people when it seems reasonable. So this approach has kind of broad applicability to really any RCT you want. So if you have randomized controlled trials that are happening and you have some sort of external real world data that you wanted to borrow, it could potentially allow you to get more precise information from like futile RCTs if you have an RCT that like just didn't have enough individuals, or potentially have more treatment effect subgroup analyses with some more precise effect estimates. My future work is like we're working to kind of extend to external real world data, because we started off with the observational data, which has these kind of inherent assumptions where there's just a complete case situation. We don't really have a lot of missingness that's happening. We have a lot of the same sites, so a lot of the same measured covariates that were kind of measured similarly. So kind of looking at how we could deal with differential missingness between the two arms is potentially kind of something that might be of importance when using real world data or electronic health records, increased missingness of covariates, differential missingness between studies, harmonization across study sources. So there are a bunch of different ways in which it might need to be extended when using something that is not quite as clean or easy to use as an observational data set that we did use. And then applying this approach to COVID-19 trials with a potential application for using the N3C data that everyone here knows way more about than I do. And then also designing a trial that allows us to borrow from external real world data for futility analyses, which would could potentially allow us to kind of call out futile treatments kind of faster and at an earlier time frame while avoiding inflating type 1 error if you again only use the RCT at the final analysis and avoid borrowing. So yeah, just wanted to thank all of the people, the University of Minnesota, the Translational Research and Career Training Program, TRACT, and all of the TRACT TL1 scholars who are wonderful, CTSI, the Clinical and Translational Science Institute, MSI, Insight Network, and then my dissertation advisors, Joe Miners and Tom Murray, who are all fantastic humans. And if you wanted to, know more, please reach out, but I'll open it up to questions, although I don't know how, I guess, this works.
Okay, well, we have different flavors of questions. So Harold, are you a panelist? Okay, so beautiful work. Sounds like steps 0, 1, and 2 are constructing a prior distribution. So 2 questions. What does the Bayesian version look like? And should you be accounting for surprise, which I think some Bayesians worry about?
Yeah, so this actually is in the Bayesian framework. I should have kind of clarified that. So we are in a Bayesian setting. So it is. So there isn't a different Bayesian version, I guess, is kind of the answer to that. And I'm unsure on what you mean by accounting for surprise. I guess that's kind of my goal with the second step, is if you have something that's inherently different kind of between the representative group and the kind of RCT that you're working with, you hopefully can account for that via this multi-source exchangeability modeling framework. That's kind of the goal with that, where you are using kind of some of the information at your disposal.
All right, no, thanks. And then Yi. Oh, excuse me. Did somebody say something?
I saw, did someone?
Okay, could you talk more? This is Yi. Could you talk more about how you calculate the similarity between observation data and RCT data? How this determines the weight of the observation data?
Yeah, so let's go to kind of here. I think this is kind of the major component where we're talking about that. So the kind of propensity score is really the step where we're taking kind of all of our X covariates or all of our baseline kind of covariates and determining this summary measure or this propensity score, which can kind of be thought of as a summary measure of the degree of similarity between the RCT and the observational data. So that's kind of the big step. So if you have propensity scores that are very different between the observational data and the RCT, then you're only pulling maybe like a very small fraction of people who are have propensity scores that are similar enough to even pull. But if they're entirely non-overlapping, then you would have the case where your propensity scores, you wouldn't be able to borrow kind of anyone from that propensity score step. Was that kind of hopefully answering the question?
I have a follow-up question, unless you, sorry, did I cut you off?
Oh, no, you're good. I was just I'm never sure. Okay, sounds great. Go for it.
Yeah, so I was just thinking, so one of the things I've been really, really excited about, and I just love your work, beautiful work. Thank you so much for presenting today. I was wondering, I've been really kind of excited about the idea that N3C, as a very large cohort, could potentially be used in combination for trials that have more pragmatic or dynamic arms. And how do we basically do that propensity matching, but during the course of the trial, right? So that we can rejigger our arms as we go, recognizing that we have, this incredible, richness of data, even though it's messy and has all the issues that you've just laid out. So do you think that that's a possibility for the next steps for what your methodology is showing here?
Yeah, so that's kind of, there's some things that we're kind of working on is like designing a trial with this kind of approach in mind, where we can figure out like how much we should borrow as we go. The big thing that kind of comes into question is if you're now designing a trial and you are trying to borrow from something and you have an interim analysis and you assume that you're borrowing X percentage or a certain amount from the real world data into your control arm, how do you make sure that it's the same amount of borrowing at the final analysis? Because you could get in a situation where all of a sudden at your final analysis, something has shifted and you're no longer going to borrow the same amount from the real world data at your final analysis as you thought. And then if you had randomized more people to your treatment arm because you were like, we got plenty in our control arm, but you don't have as many as you thought at the final analysis, trying to figure out how to kind of make sure that that situation doesn't happen is kind of a big thing that I'm thinking about too. But yeah, for the propensity scores, I wonder if like we could work on like, they could potentially be updated based on if we find kind of new prognostic covariates that we think should be included, or if we find that maybe there's a group of people that you don't need it, they're actually not, like that covariate isn't actually making a difference from an outcome level. So that would be an interesting framework for sure. Yeah.
So I was wondering a couple things. One is certainty quantification or getting, error estimates. So one of the things about N3C is as you could see, that it's an ocean of data.
Mm-hmm.
So you can draw multiple populations in propensity match with completely separate randomly drawn populations, or you could add whatever sort of spin you want. So if you have the variables you're matching, and then you have the variables that you're not, or even if you have the variables you're matching, but there's missingness in the N3C data set, it seems to me that there were a lot of things that could be done. I mean, naively, I think in terms of a Monte Carlo sort of analysis, but I'm sure people like you and Harold and can be much more sophisticated. But so maybe, what do you think with 4 million COVID positive patients, you have an ocean of potential data. What are your thoughts about what could be done to actually deal with the issue of the unknown missing, things that you don't really know are going to impact results?
Yeah, I mean, I think there's so much there. I think what seems like super exciting about the fact that there is so much data is hopefully you can like kind of figure out like which groups are actually similar, or how many kind of like figure out multiple kind of populations or draws from this ocean of data that could be similar to the RCT to kind of ID which ones are similar. This almost makes me think of identifying kind of a historical control group that I like read a paper about recently, and I can't remember who wrote it, where they did it in like a machine learning framework where they kind of left one out for the external data sources, figured out which ones were most similar, and then kind of pulled them in. And it feels like that almost might be like a really nice way that we could do that, is trying to figure out kind of maybe different draws and then which ones seem the most reasonable based on like almost like a leave one out framework. I don't know. That's something that I just kind of think.
Yeah, I mean, one of the things that would be cool about this is that really propensity matching for civilians makes some of us a little nervous. I mean, we think about the extraordinarily excellent propensity match controls amongst the hospice population, for instance. I mean, so what are you not seeing? And certainly the ocean of data where you at least have the fact that you have a little bit of everything certainly would, I think, mitigate some of these concerns. Okay, so and Harold says, in other words, use a slated RCT as the base RCT. And Don Vessel says, in the context of a clinical trial, effectively communicating these cutting edge statistical approaches and their complexities to a DSMB would be a fun challenge. Yeah, what's a challenge like that? I'm glad I'm not doing this. So Okay, this is wonderful. Any other thoughts, comments, questions?
I think I'm trying to keep track of everything and I'm thanks, Jane. Oh, it looks like how much unmeasured confounding in relation.
Did we, okay, applying this approach to a use case, how much unmeasured confounding, e.g. e-value in relation to the expected effect size, is there after propensity score matching between the treatment arms?
So I haven't computed any sort of like actual kind of metric for this, but I think hopefully this will help kind of answer this question in general. So with that this kind of second step, after we kind of pull out the people who are similar to the RCT and look to incorporate them, the multi-source exchangeability modeling step will hopefully mitigate kind of this unobserved or unmeasured confounding by using the outcome information at our disposal to figure out how much we should actually borrow. Because hopefully, if there is unmeasured confounding, it is kind of impacting your outcome in some way. It probably would be impacting your outcome in some way. And so we would use this kind of degree of similarity, this kind of function to update how much we weight the use of the kind of external data versus not using the external data kind of components of the posterior for the event rate probability. I don't know if that helps answer it.
Yeah. Oh, and Harold asks you to please post your email address again.
Oh yeah, definitely. I'll go back to the end. There we go. Yeah, so Hayne 108.
Would that be?
Oh yeah, that's a good call. Great idea. I'll add that to the chat. I have somehow the chat open and not open all at the same time. But yeah, thank you all so much for having me, by the way.
Well, thank you. I really
I didn't say that.
You know, this is very thought-provoking, really interesting.
Yeah, these are all really good questions.
And hopefully we'll get you involved in some of this. This
Yeah.
is be great.
Yeah, thank you so much. I love the N3C data source. It seems like amazing. It's such a fantastic kind of process that everyone did. So thank you all for that.
Great. Okay, we'll give you 15 minutes back, and thanks, and we'll see you next week. Take care. And thanks for coming.
Bye, everybody. Thank you, Lillian.